Introduction of biomaterials, tissue engineering and regenerative medicine, and what's the global demand of biomaterials, and developing commercialization route for sustained production and delivery of therapy. Key developing commercialization route for biomaterials, because manufacturing, developing biomaterial is good, but their commercialization is the real challenge. That I will be talking about. What's the way forward? What we can, how it can be, can bring this into the clinic, from lab to the clinic. I will be talking about what's the spinning of the university's best model. And our proposed model that we have established back in 2012, and still models there I will talk about. What's the situation of burn and diabetic foot ulcers worldwide, and what's the existing treatment option in developing countries like we have in Pakistan and Sri Lanka, and our proposed ideas for synthetic skin substitutes and advanced wound care products that we have been working for the last 7-8 years. And our commercialization strategies and recent licenses that we have already licensed to the industries. What's the biomaterials, tissue engineering, and regenerative medicine is? This research, this slide will talk about the introduction, brief introductions. That the biomaterials are basically for bone repair, for example, implants, screws, and fillers. For skin repair, we need graft. For dental repairs, for fillings and implants. When the tooth is decayed, so the implant can be comes in and can be used there. And the other applications are, for example, hearts and sutures, wound dressings. That's the other area. So just to flavor that what the biomaterials are. Tissue engineering, the use of biomaterials, for example, for the repair of the tissue, for example, bone, skin, and teeth. That's called basically tissue engineering. And linking biomaterials with cells, using cells alone, or applying in biological environments, that's come under tissue engineering. If we look at the global market of biomaterials worldwide, in 2017 it was 83 billion, and it is expected to raise to the 151 billion in 2021. It means there is a huge demand in this field, and there is a good business opportunity. So this slide shows that what was the different biomaterials and products we are needing. For example, if we look at the orthopedics, that's the highest demand. That's the $30 billion US demand in 2017. If we, the area what I am working is, that is the wound care. That's again $8 billion US. That's the huge market. That's the reason we have been in this area working. If we look at the economy of Pakistan, in the last decade, there has been a 4-fold increase in international publication. That's our government, and is really thinking about this to support the scientists and the universities. More infrastructures are coming along to produce the higher number of good publications. That's the reports from Thomson Reuters, where what says statistics were better than the BRIC countries. That BRIC is Brazil, Russia, India, and China. So its progress overall Pakistan is better than this. That's the graph. They took this from Nature's website, which shows that Pakistan is top of the countries which highest rise in growth research outputs, which is really obvious, and it's good for Pakistan as well. So based on this, what I can very confidently say is innovation is not a problem for the academics at the moment, but the exploitation of innovation is a problem. So we need to transfer technology to the industries, where the universities can take this technology and work on that onwards. So, for example, need startup companies at universities. That's the Western model. That's the Australian model and USA models, where the university have their spin-outs. They will be in the university for a certain period of time, and after this they go out and sell to the big companies. That's the way forward. So back to the tissue engineering, just to flavor that what tissue engineering is the use of combination of cells for engineering material methods. So it means, so for tissue engineering we need chemists, physics, we need material scientists, we need biologists, microbiologists, and cell biologists. So that's the whole combination of the team we need for tissue engineering. That's the example, basically. For example, for any specific organ repair or regeneration, what you can do is we can take specific cells from the patients, and then you isolate these cells from the patient, and then bring this to the lab, and then cell proliferation will happen, and then you need a scaffold. That's the biomaterials comes in. You need a template where the cells have the area, where they have the space, they can grow in, they can come. They need some physical support for the cells where they can attach. So that's the biomaterials come in, and then you put your cells on top of it and construct them in the bioreactor under specific conditions that the implant will be ready, and then you can put back onto the patient. That is tissue engineering. So what's the 3 tools of tissue engineering? We need very specific cells. That's really important, because cells is the most intelligent part of the body. They convert into the different organs and specific things. And you need scaffolds, biomaterials that provide support and shape to the constructs. That's basically decide that what sort of the shape you will be required. And then there is the cell signals. You need some growth factors which can really help these cells to grow very well. So what's the need for replacement, for example, biomaterials? Why we are putting so much efforts in this area? For example, there are 3 million procedures per year of the skin. That's a huge number. And bone, 1 million procedures per year, cartilage, 1 million, blood vessels, 1 million, kidney, 600,000. Again, the number is really high. And the liver, 200,000 procedures per year, and nerves, 200,000 per year. So that's the area where the scientists have to really think about. So that's the future of scientists, basically. What's the biomaterials? Substance that act as a whole or part of a system that is used for the repair or replace of the tissue that has been gone off, that has been damaged, or organ of the body. So that's the simple definition of biomaterials. So what biomaterials do? What biomaterials doing are they create a biomolecular and spatial environments that is needed for cell proliferation and vascularization in tissue engineering and regenerative medicine. That's the impact of tissue engineering. Here what you can see, that's the hair that has been 3D printed in the lab. So they put some cells on it in the biological environment, in the lab environment. When this is grown, you can put back. So that's the really good success. So what do we want in a scaffold? The scaffold should be biocompatible, it should be biodegradable, chemical and mechanical. It should be of reasonable strength. For example, talk about the skin grafts. So it's not to be a fragile. So what surgeons would like to do is they will put on the wound and they would like to suture it. So if the suturing is not possible, then it means you need more mechanical strength, or you need more other polymer. And you need proper architecture as well. That means you need space inside. That's for example, the oxygen can come in if you talk about skin grafts. And the cells can go inside this specific pore size. If the pore size is too small, cell size is bigger, they will not go inside. So you need some specific size materials. That's the role of scaffolds, present a surface or structure that closely resembles the extracellular matrix. It means you need to develop something which is so flexible that it is close to your human body. Surfaces that could maximum favorable biological response, that's the basically desired properties of a scaffold. So that's the simple classification of biomaterials. There are natural, synthetic, or biocomposite biomaterials. So let's see what these are. Natural, for example, protein-based, collagen. So it's very famous, very popular biomaterials in tissue engineering, regenerative medicine. So most of the skin grafts which are coming along, they're collagen-based, and collagen is being used in other biomaterials as well, very frequently. So polysaccharides, everybody knows that cellulose goes. There will be, whenever there are wound at home or during the accidents, the first thing comes in is cellulose gauze. That's against biomaterials. And it's chitosan. Our group is really working on this, and that's again another polysaccharide-based biomaterial. Decellularized tissue-derived biomaterials that, for example, is from the dead body. You can get this skin and then remove all the cells and decellularize it, and you can save it and then put on the wounds whenever this is required. So that's contained the blood vessels, intestines, other species, other parts as well. So in the synthetic, it comes like polymers. There's different polymers: PLGA, polylactic acid, polycaprolactones. These are synthetic polymers. Depending on where you want to use, you use these polymers. And in ceramics biomaterials, hydroxyapatite is the very famous one, and bioactive glass as well. So composite biomaterials is a material composed of one or two or more species, and one is originated from naturally derived. So that becomes biocomposite. That's the definitions that what sort of biomaterials are existing. So what we want from biomaterials, the characteristics of biomaterials: bioinertness and bioactivity and chemical and mechanical stability, And this should be biodegradable on whatever the desirable level is, and biofunctionality and biostability and biotolerance. These are the characteristics that we are looking for from the biomaterials. This slide shows that what is the significant applications of tissue engineering so far in biomaterials in tissue engineering and regenerative medicine. For example, skin tissue engineering, tracheal airway tissue engineering, bladder and urethra tissue engineering, cardiovascular, spinal cord injury, bone tissue engineering, vascular. These are some of the leading examples that are where the tissue engineering has really been used in the clinic. That's the example. I took this from Professor Sheila McNeil's paper, where they have developed this mice skin, and this was tested on the burn patients. And what's the A we can see is the wound healed by using patients on keratinofibroblasts grown in the laboratory. They took patients' cells, so keratinocytes and fibroblasts were grown in the lab, and then this graft was put back on the patients, and it was healed very well. That's the tracheal or airway tissue engineering, where they can grow this tracheal system in the lab, and when this is grown, they can put back into the patients. So Professor Antintrell, a very, very famous professor, he is working at Wake Forest Institute for Regenerative Medicine. Luckily, one of my PhD students is working in his group at the moment as well. So he's really famous. So what he has done is he has developed this bladder in the lab. So what this is, basically this is a scaffold. He's taking patient cells grown into the lab and implant back into the patients, and it's working really good, nicely. That's the A shape. That's basically scaffolds here that he has developed. And what this B is, tissue engineering bladder, astronomized to the native bladder. It means they have connected these blood vessels and nerves together as well. And by using these polyglycolic sutures, dissolvable sutures. And what the C shows is implant covered with fibrin glue and omentum. So put some glue, that's in place. So that's the beauty of tissue engineering. So you have scaffold, you have put the cells, bladder is grown outside in the laboratory, and then implanted into the patients. That's the really help in this area. So that's the clinical and preclinical trials for tissue engineering products. This is tissue engineering urethra. That is a clinical study going on, and this is cellulized diameter blood vessels, a preclinical study. So again, it means the blood vessels are able to grow in the labs as well, and then whenever they are required, they could be put back. There are some other applications of biomaterials. It is a hip implant metallic is very famous. When this has been broken, they can put it be black, and the people can walk again. And this knee implants, it is very popular. So most people have it. And then in the area of the eye, it could be artificial cornea and intracellular lenses as well. So that could be the biomaterials as well. So that's the dental region basically. The dental applications of biomaterial includes the endosteal root form dental implant. That's the metallic implants can go into the bone, and it's fixed. And biomaterials tooth comes as well. That's the other area. Yeah. So that's the breast implant biomaterials. That's the again very popular biomaterial being used worldwide now. So yeah. So in the wound care products, we have synthetic wound dressing, the BioBranis product, and the Integra, very famous, developed by American physicians back in 1978, FDA approved in 1998. It's very popular being used product worldwide. Oh, okay. What's the challenge for biomaterials used in tissue engineering then? So we have the main challenges are the acute toxicity, genicity, neurotoxicity, immunogenicity, immunotoxicity, carcinogenicity, endotoxin. So all of these tests have to pass biomaterial successfully before it can be used in the human body. So it needs a lot of extensive toxicological studies before it is placed on the human for the first time. What's the future directions in biomaterials? So what's the suggestions? So how we can expand in this area? How can make this area more useful so that we can provide better healthcare solutions for the patients? So what biomaterial scientists and engineers should increase the vicinity of application by integrations. It means they should have more collaboration, more productive collaborations of biomaterials with molecular biologists, biochemists, genetics, physics, and other area of the science. It means that you need interdisciplinary research before you can develop more biomaterials. So regenerative medicine challenge. So whenever there is a challenge, it means we have the opportunity to solve the challenge. That's the academics would love to work on the challenge. So design of biomaterial that function in the body, that's really important. For example, for the skin products, we need somebody for the full thickness burns. We will need something which will be dissolved quickly in weeks time. For bone, we need something which can stay there for longer. And similarly, some other applications, we need something permanently there. So we need to decide which sort of biomaterials is needed. And getting NF cells and cell engineering and tissue engineering, that cells should be happy on your material, basically on your biomaterials. And vascularity, that is the new blood vessels formations. For whenever there is a tissue graft, so and put on the body, so the blood vessels should come in. So what do blood vessels are? They bring oxygens, they bring food for the cells. That's how the cells becomes activated and tissue engineering starts. And cost of tissue engineering development procedures. So if it's really expensive, forget us about it. Nobody could be able to use it. It should be affordable. That's the other thing. Sustainability. So working in the working models for developing countries are, for example, there should be something which is sustainable. For example, if the companies can make it for like 2 or 3 years, and even after that, if it is not sustainable, so no need to develop it. So you need to think about the biomaterials which are sustainable and can go longer. So that's the slides basically shows that when you are developing projects in the area of biomaterials, tissue engineering, and regenerative medicine, what are the key factors you have to think about? So scientists is here understanding the patient's needs, and you need to go and talk to the clinicians that what's your problems are. And then when the problem is in hearts, you have to come back to your laboratory, and you have to designing and producing biomaterials at the lab level. And then you have to testing of these material in relevant animal models. When this has been tested in animal models, you have to go and talk to the regulators. That will read your dossiers, very detailed dossiers, and they will say they will send you to third-party toxicity trials where they will be testing your material very extensively, and then they will get first-in-man safety study. Usually, this is on small number of patients. And then when this is successfully gone, first-in-man safety study, then you go into larger clinical study, and after this is the industrial production. That's the area of biomaterials where you need the teamwork. You need surgeons, you need cell biologists, you need material scientists, you need immunologists and animal researchers, regulators, you need industry support as well, and much more as well. So in this is the area where alone you cannot work. You need a lot of collaborations. So you need, when you're working in this area, you should be able to know that how you will convert this technology into business. That's good for your national economy, and that's something academics should take interest as well. And they have to rightly at the beginning they have to see that what's their model. So these are the key questions which academics have to think about. Will the technology you are developing be affordable at the point of use? How will you fund the developmental cost, from where funding will be coming, and who will you fund the routine use of biomaterials? Commercialization is via company or licensing to a company, startup or spin-out. We will be talking in details in coming slides. What will be the cost of the biomaterials? Do you understanding economics of introducing new materials, or you will be hiring a relevant person who understands market dynamics? Safety is important. What are you going to do to convince the regulator and clinicians that your material, how you will convince the clinicians your material is safe, and patients as well. So for this, you need in vitro test, in-house animal testing, contract testing services, and you need to follow the ISO, ASTM, and FDA rules and regulations. So what's the standard? Standard is a measure of product or data quality and consistency, and is valuable for customer advice, confidence. You can say that we have followed this standard. And regulator confidence and vendors' abilities. It means when in this area you have to follow the certain standards, and standards also facilitate trade, help establish emerging technologies. It means it can really help to grow your business as well. So intellectual property is really important. That's the intellectual property is the patents so you need to get, if you think this is something really important, then you should think about patents, because because maybe there's no business without the patents. You need to get the patents. You have to think about that. How will you develop your research such that it becomes a tractor for commercialization? That you need to have. You should have patent, because when you are talking to the investor company, you can show that this is the patent. It's protected. This is our technology, and which is exploitable. Why would a commercial company invest in your solution? For example, that's the question. Process of filing the patents, you have to understand that who will be filing. Your university will be filing, you will be filing, or your industry will be filing. So this is really encouraging slides that creating commercial value from biomaterials from the research which has been happening at universities. For example, this is example from the University of Utah. So this Center for Therapeutic Biomaterials, they have this Centrex company. Under this, they have 3 companies: Carbaline Biosurgery, and then Glycosan Biosystem, Centrex, and then there is a Biotime. So that's the university-based research has all these potential. That sort of things need to adopt. So this is the commercialization model which we have developed back in 2012, and still we are looking at this, and we are trying to adopt this, and we are working that how we can bring our research from lab to the clinic. That's we are on the top. That's the tissue engineering lab, and below there are plastic surgeons, and after this we need animal trials, third-party toxicity trials, and then approval from the regulatory approval. So these are all these things. When you are working in this field, you have to think about where you will be sending for third-party toxicity trials, whether you will decide, your regulator will decide, or the approval from the regulatory approval. And then, for example, your artificial skin is ready, how you will be sending this to the patients, how patients will get it. There are 2 ways which we think are workable. For example, if you can have a skin bank which is non-profitable and hospitals demands, you can send skin to the hospital. So the other thing is, if you are working with lead clinicians, which are very popular, then people trust it, and working in very good hospitals, if they say that this skin is good, they endorse it. So the industry companies will come definitely for manufacturing, and they will be manufacturing, and you can commercialize. That's basically our model. So how to spin out the university-based research? So this slide is about talk about that what you need. For example, pre-spin out. So pre-spin out, you need intellectual property. You need to talk to the professor, or if it's your own, then that's fine. Yet and that's your pre-spin out. One of the requirements that you have secured the IP. IP is in hand. Patents have been filed, and developer looking forward-looking IP strategy as well, and identify what license to and copyrights, patents, all these stuff. This is pre-requirements. It means you should have patent in hand. And then you have spin out. So for this you have licensing agreement, patent sale, or setting up a company. For example, what you will do. So for example, if you are setting up a company, you need a funding, and you need to think about from where this funding will come, and then you have to think about the growth, that's the expansion of your spin out. And after certain times, people would like to exit this. Means once this is working for 3-4 years successfully, they would like to sell it to the bigger companies. So setting up a company has following assurances have to be made in this regard before you are planning, for example, to set up a company. So what you need is company has access to the required IP, and is structured in a way that appropriate for future investors, does not have any tax obligations for academics, because if the IP belongs to the academic and this is licensed to the company, so don't put tax pressure back on the academics who has invented this technology. That sort of things. Ensure everyone is fairly getting incentive for the work which the people are involved in. Company may have independent advisors as well. These are basically the people who have already in this field for a longer time. They could be regulatory angle advisors. They could be manufacturing advisors. and according to the need of the company. So from where this funding will come, may come from grants and translational funds. Most of the countries have really grants for startups, where they're supporting new startups. And the proof of concept funding, friends and families, if you're really nice and if the people, your friends and family trust you, they may give you some money to start up the company. And you can go to the angel investors and venture capitals. So what's the angels and capital investors are? Angel investors are usually individual rich man. So basically this is an individual, and usually they are doing some other full-time job, and they invest in the long term, and once they bring product to the markets, they can earn some money. So on the other side, the capital ventures are, that is a company and invest as full-time job and wants a board seat as well. They would be part of your company as well, and wants larger returns on investments. That's the venture capitals. That's the things you have to think about from where you're going. Now back to our research, that what we have done in this area in the field of biomaterials, tissue engineering, and regenerative medicine. We know that skin defects are caused by burns, diabetic foot ulcers. That's the extreme. So for example, if we look at 2006 in WHO report, 265,000 deaths every year are caused by burns, and most of them are happening in low and middle income countries. That's really sad. So it means the scientists sitting in these countries have to really think about how to decrease this number. There are more than 14,000 major burn patients per year in Pakistan, and similarly in other developing countries as well. So these numbers are higher. So it means there is an urgent need for functional and cost-effective skin substitutes which can help to heal these wounds. So if we think about what's already available in the market, yes, Integra is available in developed countries. It's being used for many years now, successful there. So what's the Integra? Integra is basically collagen-based matrix, and on top of it, this is silicone membrane. It's a bilayer membrane. So it's very expensive. For example, in Pakistan, talking to the clinicians, most of the clinicians, they give prices like 4x4 centimeter patch is like $800 US. Think about the hand surgery, how much it will cost, like $10,000 US. This is very expensive, not affordable. So it means we have to develop something indigenous in these countries to support our patients. Cadaver skin is being used in UK, but not that popular. But in our countries, due to ethical reasons, religious reasons, it's not very common. What's the autografting? That's basically, if your one leg is burned, you can take skin from the other legs, spread it on, stretch it, and then put it back. Again, this is okay if the area is small. Total body surface area which has been burned is small. If it is more than 30%, it means there is a problem. You will be causing infection everywhere. The area which is burned can cause infection, and the area from where you will take the tissue, that will cause the infection as well. So in that's the case, we really need skin substitutes to apply. So that's the angiogenesis. Angiogenesis is formation of new blood vessels. There has been a lot of grafts scientists and industries have introduced into the market. When they were placed on the wound, they couldn't make this wound back quickly. So it means they couldn't help to generate the new blood vessels formation. Think about if it's a wound, if we put the graft, so the blood vessels should come in 3 days' time. So if they are not coming in 3 days' time, so it means they will cause the infection. You remove the graft, and the procedure starts again. So, but if you put the graft, and if it's taking blood vessels in 3 days' time, so it means this is working nicely. Then it's growing, and the blood is coming, food is coming, oxygen is coming, so the tissue healing is going well. So we are working in this area that we were trying to develop the skin substitutes, dressings, which can support new blood vessels formation. So looking at the diabetic foot ulcer cases in Pakistan at the moment, there is more than 5.2 million people with diabetes. 15% will get diabetes, and 15% will get diabetic foot ulcer, and out of these, this number is 8 to 21, but some reports says 25% goes on amputation, that the cutting of the leg or something. It's very sad. Even in UK in last year, there was a report, UK is a developed country, amputation rates, 15 amputations per day, even in UK. So there is a huge demand of these grafts, which can be used to heal these non-healing wounds. That's the classical approach. That's the debridement. If the patient comes with diabetes, what they do is they remove all the dead tissues, and they will do it like that. That's the old way, classical way, but it's not really useful. What's the other way? The other way is tissue engineering. For example, that's the My Skin developed by Professor Sheila McNeil, and this has been put on this ulcer, and this was healed properly. And that's the other example where this another dermagraft has been placed, and this wound has been healed very well. So it means we need to develop advanced wound care products. That's our strategy, that how we are exploiting our pro-angiogenic materials. What's the pro-angiogenic materials? The materials which support new blood vessels formations very quickly and helping wound closures quickly. So the coming slides I will be talking about that what we have achieved in this area. So developing of the pro-angiogenic, this is just one example. For example, if we get cut in our hand or body, so how our body is responding is, in our body there is a VGS, just called vascular endothelial growth factors, that comes in and supports new blood vessels formation. This is just example, that is untreated group. No, we can see some blood vessels naturally growing in CAM assay. This is chicken fertilized egg. And where they have placed this VGF, 200 nanograms, we can see there is a massive vasculature, it's a small blood vessels growing. But the question is VGS practical? There was chances it was practical. That's the reason. Scientists has been investigating this, and the industries were jumped into this one too. They have introduced some products based on VGF, but they are no more in use, because this is very expensive. 500 microgram is like $830 US. Expensive. So you need certain conditions to make it stabilize. And for example, the shipment is a problem. So all these problems is could not make it like a practical use. But still we need something which is stable, inexpensive, and is working as good as VGF. So that's basically our group is working to develop something. So we have been working in this area, and we have worked on quite few pro-angiogenic agents I will be talking about. So for example, heparin. Surgeons have been using heparin for wound healing for many years. But what we are doing is we are putting this heparin into the grafts, making it implantable materials. So that's the example where we have chitosan, PCL, and PVA based membrane, put some heparin, and then we have produced this highly porous membranes, the pores we need that cells can go inside. And this has supported a really good number of blood vessels. Next slide I will be talking about what sort of blood vessels formations we have observed from here. This is another example where we have cross-linked chitosan with PVA, and then again was coated with heparin. And for heparin staining, we have this toluidine blue staining, which shows purple color when it reacts with heparin. So we have confirmed the heparin is there. When we have put this in CAM assay, we can see that blood vessels were really grown into our materials. So that's our confirmation that heparin is working fine. So that's the thyroxine now. So during my postdoc at Japan, I was working on thyroid hormones derivatives, and I was really encouraged from that stuff whether can we use thyroxine as a pro-angiogenic agent. So what we have done is we have chitosan and collagen membranes loaded with 1 milligram or 10 milligram of thyroxine. When we put this in the CAM assay, so we can clearly see that the number of blood vessels have gone very high when we have used 1 milligram as compared to the 10 milligram was a bit toxic, but 1 milligram is fine, and the number of blood vessels were increased very well. Yeah, this was the time when we took these grafts to the plastic surgeon, Professor Mustahsan Bashir. He's one of the leading plastic surgeons in Pakistan, and when we showed this, he was really interested in to test these materials on the animals, and he was interested in to do these experiments with him. And we have designed this animal trials experiment with him. So on the day 0, you can see that we have made this full thickness wound in rat, and that we have sutured this simple chitosan, and this was chitosan with thyroxine. So on the day 4, what we have seen is from this middle row, what we can see is not much information. So only we can see is this attached very well to the wound. So that surgeon working with us was really curious to see that what is underneath. What he's done is he's make a little cut, and then this was left up, and with the back torchlight, what we have seen is where there was thoraxin, there is really clear circular blood vessels that has gone into the area where there was a thoraxin-based material was present. So it means thoraxin was really convincing cells, endothelial cells, to come in and make the blood vessels. And just in 3 days, just 4 days time, look at how many blood vessels has been. That sort of activity we want from the biomaterials, where we can see that the wound clear will be fast. And there was number of blood vessels was really high as compared to the material where we haven't got any thyroxine. We have licensed this technology to the Cottoncraft Private Limited. That's a company based in Lahore, and they have submitted dossiers for production of such materials to our regulators in Pakistan, and they are waiting for the approvals before they can start manufacturing. That's the hemostats. Hemostats are the materials which can really help to stop your bleeding, because a lot of deaths worldwide happens due to the extensive blood loss. So we have established this technology, the product that is called Cytostat. So if we apply this to the bleeding, it can stop bleeding very instantly. So again, Cottoncraft has they prepared this dossier, because we have licensed this technology to the company. They have prepared the dossier to our regulators, so for the certain approvals. So these approvals are in progress. Now I will really pleased to talk about the collaborative project with Open University Sri Lanka and Professor Guneharath and his team. So we have been successful to win this PSF and NSF grant. And under this we have 2 teams, Pakistan team and Sri Lankan team. In Pakistan team, myself, Dr. Shehzman Safi, assistant professor, and he's a molecular and cell biologist. Mr. Ahmed, he's a research assistant in this project, and Sri Lankan team, Professor Guneharath and Dr. Khitsri Hector Jayawardena, and Dr. Chandri Rana Singhe and Priyantha. So this is the team from Sri Lankan Times. Very efficient, very hardworking team. Really, I'm really pleased to have this team with us for the successful completion of this project. So what we are doing is we are using chitosan as a carrier, and we are using fresh aloe vera gel and mixing it, and we are using ofloxacin as an antibacterial drug, and we are making this ofloxacin-based chitosan aloe vera membranes. We are really pleased to see these are transparent membranes. Surgeons would really love to have these transparent membranes, because when they will put this on the wound, they can really see what is happening underneath. So that's we achieved so far. That's the canvas, very popular in our group. We are using it very regularly for our pass-fail criteria, whether our materials are helping blood vessels or not. Based on this, we will see whether we want to go into the further detailed animal trials or not before this. So this is one of the really good assays. So what we have seen is that that's the first is chitosan simple, that's chitosan aloe vera. So we have clearly seen adding aloe vera, this is bringing a lot of strength to this, and the number of blood vessels has been enhanced. And adding a small amount of the sugar is useful, but the higher concentration of the sugar is not really effective. The blood vessels are decreasing. It means the low concentrations is the way forward. That's the again slides. For example, we have untreated group, VGF. We want to develop something which is really we're behaving like VGF, stable and cheap. That's the area that we want to develop something. So look at this slide. That's why my work. I've been doing this in 2014 in University of Sheffield, where something came into my mind based on my previous knowledge that maybe this could be the molecule which is stable, inexpensive, and this may behave like VGF. So look at this slide. That's on left is my control, on right is our angiogenic electrospun fibers. Look at this. That's where these blue arrows are heading. So this is a clear new blood vessels formations, tiny ones, when we put this small organic molecules as compared to the control. There are not many. So this was really interesting, promising time that we should really investigate in this, and we have started thinking about exploiting this further and also patenting as well, because this is something not obvious, that we have developed something which is behaving like vascular endothelial growth factors. So what this magic molecule is, this is 2-deoxy-D-ribose. That's a small sugar molecule. So we have joint patent with the Professor Sheila McNeil at University of Sheffield and myself at ComSearch University, Islamabad, and working at Lahore campus. So what we can do is we can release this sugar from a variety of the biomaterials, sheets, electroporcelain materials. They contain all the specific infrastructure, a microstructure, macrostructure that is porous and stuff like this. ChemAssay confirmed that this is working nicely. And animal trials shown us that at day 17, in the case where we have put this sugar-containing graft, the wound was closed very well. So we have done the histology as well. So in the histology, we can see that there is a proper formation of epidermis and dermis and even hair follicles as well. So this is detailed animal trial slides. On day 17, we can see that the sham where we haven't applied anything, that the wound is still there with scaffold, with control, with that's the simple chitosan collagen membrane. This even in the middle one is not closed, but the one with the sugar is closed very well. This is showing the strength of this technology, that wound has been closed very well. So there was a good media coverage on this. Yorkshire Post, news in the University of Sheffield. For example, Sheffield's engineers discovered some sugars that's good for you. Okay, and that's another news. Researchers stimulate blood vessel formation with sugar. But this sugar is 2-deoxyderibose sugar, and even in Outlook and Pakistan Local Express newspaper, and from India as well, and MIT Technology. So I mean, this was a really encouraging point where people have started thinking about that we have something which will help us to heal the wounds which has been not healing for a very long time. That's the work from Sir Khan's work, and what he has done is he has prepared electrospun fibers, and that's the control PHBV. We haven't got any 2-deoxyderibose. Look at the bottom. That contains 250 microgram of 2-deoxyderibose, and that's materials on my right bottom that contains 500 milligram of 2-deoxyderibose. So we can, if we look at the control and this, we can clearly see there is a massive difference between number of blood vessels. We can clearly see that 250 milligrams, a lot of blood vessels are coming. That's basically we wanted to see from this agent. Higher concentration is not good. We have been working very extensive to find out the right therapeutic range for this. At this stage, this technology was identified for huge commercial potential. So this was licensed to Canenta. That is a new spin-out startup company established in Australia. So they were really interested in to deliver this sugar from commercially available dressing. So in this case, they selected alginate. It will be used it's very commonly worldwide, even in developed countries as well. So what from this project, I hired Mariam. She's a wonderful researcher, and she managed to put 2-deoxyderibose into this one. We got sterilized dressings, open in sterilized environment, and we have sterilized solution of sugar put on this, and everything sterilized. And under that sterilized dressing were tested on the rat model as well, talking in a while. So we were really interested to see that how long this will take that the sugar is gone away from these dressings, because if we put this on the patient, if everything goes off in one hour or so, it's not useful. If everything goes off like 3 years, not useful. So we need something which goes off in the required period of time. For example, in this, sugar is going off in 8 days' time. That's really good. It means one dressing can stay there for 8 days, and then we will put the new dressing. That was the ideal time period as well, and the duration as well. So we tested this in the animals. What we have seen is that after day 22, the wounds were still there for sham and alginate, but with the sugar, they were closed very well. So, well, that's the conclusive slides, that what we have done so far with our research, that we have developed two types of the skin substitutes. That is thyro skin substitute, and I'm calling this sugar skin substitute. This thyro has been licensed to Cotton Craft for manufacturing. We have patent on this, and they are planning manufacturing after necessary approvals. And the other one has been licensed to the Canenta, that is That has been licensed. That UK patent has been filed as well, and followed by the Pakistani patent and the PCT international patent as well. Now, because this technology is with the industry, they have other interests as well, so they have started filing the regional patents as well. That's the Canenta. That's called Advanced Wound Care Biotechnology Company, established to bring affordable, effective advanced wound care products to emerging markets around the world. So that's the spin out on this. So what is the conclusion now? So what's the strong point? Universities are IP houses. University contain professors. They have researchers. They have better infrastructure as compared to the industries. They can develop technologies. And they have more power to basically get the patents, produce the patents. Yeah. So where is the challenge? Exploitation of the technology. Academics are publishing a lot of good papers, and that's it. So academics have to really think about how their research could be commercialized. That's basically the way forward. So in this is exploitation of the technology. There is no roots are existing that how to exploit this technology. And then what's the way forwards? Industry and universities need to write the joint projects. So for example, when academics will go and sit with the industries, they will think about each other's, they will think about each other and then see where the industry interest is, where the academics could be utilized very well. They can have like joint projects, and they can submit into to the other agency as well, even that the money will come and both can get the benefit out of it. Or when they will write the joint projects, and at the same time industry may get interest that this is a lot of business interest. Why not I sponsor this? The industry may sponsoring your projects. That's the way forward. And we need research and development champion from the academics, which have really great interest. They really want to develop the technology. They really want to serve their nation. They really want to have the business interest that they want to do a research which have a business interest. So we need such minds from the academics. That's the model for developing affordable biomaterials for developing countries. This is something which I have been working on these slides for a long time. And that's my thoughts, basically, the personal thoughts. What I'm thinking is university-based knowledge expertise. We have chemists, engineers, we have microbiologists. We have that's the really big bank of knowledge. And we have ISO in place. Most of the universities are already following international standards. And when most of the academics have the prototypes in hand, which they are not thinking may be useful, but they are useful. So most of the universities have prototypes in hand. What's the industrial attraction and growth? So, for example, they can see their high-tech products. For example, industries have been in business in their own area for years and years, but the developed nations, they have developed the advanced versions of the products, and they are doing better business. So universities can look at the products developed by the academics. They have the high technology. They have more importance. They can go globally. So these sort of things the industry can think about. And such products are not in the market. Yeah, that's really good interest for the industry. For example, if nothing is all in the market, company will produce it. Definitely there is a good business. And then cost-effective products, indigenous sources. The academics can think about how they can solve their problem by using their own resources which are available in the country. Trained HR available to address industrial problems and trainers. Universities have already staff, a lot of staff which knows the spectroscopy, which know the production characterization and stuff like this. And university could be R&D house for the partner industry, where the industry can just put some money and utilize all these resources. So what's the facilities at the university? Most of the universities have like fabrication lab. They have basic preparation lab is there, and cell culture labs there. SEM, XR, these all the machines they have, and they have like. They can be utilized as a staining as well. Most of the departments are there. They are doing this histology. So almost all of the industries maybe don't have this setup. They can utilize this setup based at the universities. Animal experiments they can do at the animal house in the universities. And for example, this was my case where we have diabetic rat model as well. So if the industries are working on the material which supports really healing in wounds in diabetic patients, so they can use this model. So what we are looking from the industry, that's something. That approved setup for biomaterials, for example, industries have GMP environment. We haven't got GMP environment. If we produce something in the industry, the regulatory can approve this very well. And then partners, collaboration, and external, national, and international fundings. For fundings, we need as a collaborators, and universities, industries could be a really good collaborators in this sense. So, and I would like to really acknowledge Professor Guneharath and his team and Open University Sri Lanka for inviting me here in Sri Lanka, and especially for this distinguished lecture as well. Would like to thank Higher Education Commission back in Pakistan, Pakistan Science Foundation, and NSF in Sri Lanka. Would like to acknowledge my clinical partners, Professor McNeil at University of Sheffield, and my team, and thank you very much for listening.
Dr. You had a very nice presentation
Thank you very much.
On the subject, the biomaterials and the tissue engineering. I would like to just ask a few questions, few things for discussion. Now, when we use these scaffolds or the biomaterials, are they, end of the day, are they removed from the body, or what is the usual fate?
Yeah, it's a very good question. It depends on what sort of applications we are doing. For example, if we talk about the skin, so there are two types of the materials in the skin. So some of skin substitutes, for example, and the other are the wound dressings. So in the case of the dressings, for example, if we are using cellulose or the like alginate materials, when we put this on the wounds, they will not stay there forever. So it's easy, they will come off. So the other products, for example, if there is collagen-based products, which our body can digest it very quickly, because they are biodegradable. So they will stay there for a few weeks, and the body will metabolize, and then we break it down in small pieces and will go away from the body. So again, it depends that what sort of applications we are planning. For example, hip implants. We don't want our body to dissolve it.
Right.
So we want that the hip implants stay there forever. Right. So we have to use something that is really stable for a long time. Yeah. So it depends on what sort of applications we are. Tooth filling, for example. So everybody will like to have surgeon and say, oh, lifetime filling. So sometimes we feel after a few years this is gone. So it means this is something which we are not wanting. For example, if it is like tooth filling, so it should stay forever.
So that means we have to think about different types of materials for different applications.
Yeah.
Now, you're talking about this taking certain cells and then preparing, taking to the laboratory and then preparing certain scaffolds and then bringing back to the patient. Now, how does it work? It has to be done on individual basis, right?
Yeah. So it's a very good question. So from here, I would like to share the experience of my collaborator, Professor McNeil.
Yeah, sure.
So she has started this idea because the keratinocytes, the top skin cells, they are being used for a very long time.
Yeah.
People will take it, bring it into the lab, grown on the scaffold, and put it back onto the patients. So they established this technology very well in UK. For example, what they are doing is when the patients comes to the hospital, they will send a very small biopsy back into the laboratory. Patients' own cells are coming into the laboratory. They will expand it on the tissue. So in this case, they are using like a sheet. On the sheet, they will grow it, and some sheets they will prepare and send it back to the hospital, and then they will implant these sheets back into the patient.
Right.
Yeah,
so that means if a sort of a nail or a finger is cut or maybe removed, you can take it will fill with that.
Yeah, I don't know the finger if it can be regrown, but that's
not the whole
skin,
finger.
Yes, skin, yes, for example. So if you talk about the bladder and some these stuff, yeah, they get the specific structure, and then the specific cells will come and then grow, and this can be implanted back.
Okay,
yeah.
Right. So that has to be done individual basis.
Yeah, that has to be done on the individual basis. You need patients on cells.
On site.
On cells.
So you were talking about diabetes, foot ulcers, and amputations, and so on and so forth. Now, what is the real reason for not curing these diabetic foot ulcers?
Yeah, that's the diabetes is very complicated. So, you know, when diabetes, there's a lot of dysfunctions happens in the body. So one of the problems which comes is that you get you get this wound, which is not healing, unfortunately, under ordinary treatments. What happens is there's nerves has been damaged in this area, blood vessels has been damaged in this area. So even these classical treatments are not working. What surgeons will do is they will offload the pressure. For example, if the pressure is coming, they will try to set up in a way that there is no pressure coming on the wound area. That's the classical ways. Or oxygen therapy is another way. They will put a lot of oxygen under pressure. For example, if there is a wound on the foot, they will place in a chamber, put a lot of oxygen. That can help in healing. So the challenge in this case is the blood vessels damage is the most common. That the blood is not coming there. So if this blood is not coming there, so this regeneration is not happening. So that's why we are developing these products that is really good strength to bring blood vessels inside. And
is that basically because of less pressure down from the heart, or?
It's not pressure down because these blood vessels has been closed with sugar. Yeah, these has been blocked.
Okay,
yeah,
right.
So that's the reason it's damaged. Yeah, so you need fresh blood vessels to be grown. Yeah.
Yeah. So you were talking about lots on commercialization.
Yeah, that's the area we've been doing for last couple of years, very extensively to bring these routes. Yeah.
Yeah. So. Whenever we are thinking about commercialization, I mean, you had so many models. I mean, what do you think the best? Is it, say for example, when you start with the project, I can remember when we were having, when my supervisor was doing certain projects, he used to have a collaborator from the industry at the very beginning.
Oh.
So how feasible or how difficult in that nature?
I'm here. I would like to share my personal experience sitting in Pakistan, where it's not very common that university research go into the business and the industries has been utilizing, but we have done this very well. The first thing is you academics have to really think about the patents. The first thing, don't lose your IP. Think about patent. Think about patent. The first thing is you have to secure your patent first. If you have done some research, published, that's public property. Which is again good. Yeah. Somebody will take the benefit, but you have, because this is your work. The first thing is you have to think about the patent. When the patent is in hand, so ideally, what the Western universities in USA and other developed nations universities are doing is they allow professors to have spin-out companies inside the university. There's a lot of support from the universities. The lab space could be used for the spin-out companies. A lot of HR initially instruments could be used on low price. That's all the strength come from the universities, because the universities have all these infrastructure very well established there. And this could be really good for the startup. Unfortunately, at the moment, it's not happening in this region. So this is something that's the ideal model. And also, if the professor have interest in this in the startup, so the growth chances are really good, because he has everything in mind, putting a lot of constant efforts, and the commercialization chances will increase. So. But if we cannot do this at the moment, what we can do is we have the possibility we can license our technology.
Okay.
When the patent is in hand, we can license this patent to the industries. We can license this to the investors. They can think about how they can utilize this patent. and they can think about its commercialization, and you will be getting money back in the form of different royalties. Your university will get, and they will share with the inventors.
So in your case, you are going out of the country?
Yes.
Are there good, huge companies in your country? Or?
Yes, in Pakistan, yes. Especially in our case, for example, wound care, advanced wound care dressings companies don't exist there. There exist some companies which are producing wound care basic products. So, for example, in our group, if we are developing advanced wound care products, so they are taking interest. They would like to adopt this technology, and they're really good interest. To get our technology, and so far there was a technology development fund funded by our government, Higher Education Commission, that was between the university and industry. And we have developed some dressings, and we have already licensed this to the companies. So they look forward to commercialize this after approvals.
Right. Just probably the last final question. Now, when we are looking at this drug development, now we were talking about wound healing. I mean, it's just a wound, I mean, cell migration or the wound healing assay or whatever, or the CAM assay. Now, shouldn't we think about, rather than talking about a compound which is going to be wound healing, whatever the assay we are talking about, should we think whether that compound is compound or fraction or whatever is going to enhance the production of VEGF?
Yeah, that's really important. I think end of the day there are various mechanisms in the wound healing are involved, and one of the mechanism is vascular endothelial growth factors. That's basically when vascular endothelial growth factor helps endothelial cells, and that speeds up the new blood vessels formations. So we have to really think, so we have to, while discovering this area, we have to see that what are the right agents which can support upregulation of VEGF.
Exactly.
So that's the area. For example, even in the case of 2-deoxy-D-ribose, so there's a lot of interest at the moment to understand the mechanism, that how this is working, because at end of the day, when we'll take this to the regulators, they will see, okay, well, this is enhancing new blood vessels formation, but how? Explain it. So that's sort of research we are doing at University of Sheffield.
So we may have to think about
the mechanism.
another assay method to see how this
how this is happening. So that's the other way that we can study that whether this certain agent is enhancing VEGF or not. Yes.
Okay. So thank you very much for coming over here and spending your valuable time with us in discussing all these things with me.
It's a pleasure. I mean, visiting Sri Lanka and talking to you all. I'm really happy, and thank you very much, everybody.
And nice to have you as a collaborator.
I really look forward to have fruitful outcomes for both countries from this all efforts.
Thank you very much.
Thank you.
And all ladies and gentlemen, it is my privilege to be here to deliver the formal vote of thanks. First of all, Dr. Mohammed Yah, you came all the way from Islamabad, Lahore. We must really thank you, Dr. Mohammed Yah. You are an internationally renowned scientist. Especially, you are working to produce affordable solutions for especially the developing world, so-called third world. It's very valuable research you are doing. Maybe developing world. Cutting edge of the science. So we must thank you, and also we started our distinguished lecture series from your lecture in 2020, and especially that I realized that you have experience in Western world. You did your PhD, read your PhD in Bristol, and also Middle East, King Fahd University in Saudi Arabia, and also Far East, that Nagoya University, Japan. So you have experience in various parts of the world. I mean, we are very happy indeed as the Open University of Sri Lanka to have you here for the first distinguished lecture. The series we are conducting at the Open University, especially organized by International Relations Unit in collaboration with Center for Education, Technology and Media, and also IT unit. So once again, we'll give a round of applause to appreciate the good work, very good presentation done by Dr. Ya. And also I must thank Professor K.B. Gunaherath, Emeritus Professor here. He is one of the key collaborators of the research between Pakistan and Sri Lanka. And he came all the way here and conducted very fruitful discussion. We must thank Professor K.B. Gunaherath, and also Dr. Kitsiri Jayawardena gave a marvelous introduction to Dr. Ya, and also all the members participated here, and especially the members from IT unit. So this lecture live streamed, so from anywhere of the world that anyone could watch, and also the recorded lecture we upload into our official YouTube, Open YouTube, soon. And staff of the Center for Education, Technology and Media, the large number of staff involved in this process. And finally, I thank all the participants here. Thank you very much. Good day.